The overall goal of the following experiment is to develop a sulfidogenic sludge from marine sediments in a UASB reactor and to evaluate its performance on reduction of trichloroethylene or TCE. This is achieved by collecting marine sediments to obtain a pool of a great variety of microorganisms that are enriched in sulfate-reducing bacteria when in an environment of the appropriate mineral medium, vitamins, and volatile fatty acids to serve as electron donors. As a second step, the marine sediments are used as the inoculum in a UASB reactor, which is set and maintained under sulfate-reducing conditions for several weeks until it reaches steady sulfate-reducing activity. Next, the consortium in the sulfidogenic UASB reactor is evaluated for TCE reduction in order to assess the capability of the sludge to transform organic pollutants while sulfidogenesis is still active. The results show that sulfidogenesis was established in the bioreactor and that the sludge was able to reduce TCE under sulfate-reducing conditions, based on the analysis of the microbial community, suggesting that the reduction of TCE was carried out in a consortium formed by sulfate-reducing and fermentative bacteria.
The main advantage of this procedure over existing methods, like adaptation of methanogenic sludge to sulfidogenesis, is that the sludge obtained is tolerant to higher sulfite concentrations and it does not present competition for substrate with methanogens. Generally, individuals new to this method will struggle because they expect immediate